A study by the Autoimmune Association found that patients typically consult nearly 5 doctors over 4.6 years before receiving an accurate autoimmune disease diagnosis. In this video, we will learn about a new technology that could solve this problem. My name is Dr. Matthew Menendez, and today I'm going to explain to you the science behind a groundbreaking paper that recently was published in the Journal of Science. This innovative study provides a compelling proof of principle method for identifying individuals who had either lupus, type 1 diabetes, severe form of COVID-19, were HIV positive, or had recently received a flu vaccine, so 5 different immunological states. They did this by sequencing genes from the blood, and they provided those gene sequences to machine learning AI framework, which proved to be 99% accurate at identifying which of the participants had which one of the 5 different immunological states. In fact, this study we think has the potential to transform the way medical conditions, for example, those that are highlighted in green, are diagnosed. But that being said, there are many promising studies that turn out to be just a flash in the pan, and only time will tell if this is one of those studies or if it will transform the way autoimmune disease conditions and infection-associated chronic conditions are diagnosed in the coming decades. Now, to understand this discovery, I'm going to tell you the story of how T cells develop and mature in your body, because this is important to understand what genes from the blood were sequenced and why AI was needed. It all starts in the thymus, the organ from which T cells derive their name. Inside of the thymus is a cell type that is by far the coolest cell in the human body, and they are called medullary thymic epithelial cells. Now, these cells have the unique ability to express every protein found in the human body. So these cells will express proteins found in these organs, but not only in these organs and other organs that are not pictured here, but also the various different cell types that make up these organs. And each one of these cell types expresses unique proteins. So if you think about how many different proteins are being made by this one single cell type in the thymus, it's a staggering number. Here's what happens below the cell surface. The genetic code is transcribed and translated into a protein. And again, this could be a protein that is expressed by cells in the brain, such as astrocytes, or a protein that is exclusively made by cardiomyocytes in the heart, and so on. Next, these proteins are fragmented into smaller amino acid pieces that are called peptides. Next, these proteins are fragmented. These peptides are loaded onto a molecule called HLA, or human leukocyte antigen. Once HLA is loaded with a peptide, it traffics to the cell surface where it is presented to T cells that are in the thymus. The T cells use their T cell receptor to physically interact with both the peptide and the HLA molecule in combination. And from here, one of two things will happen. The T cells use their T cell receptor to physically interact with both the peptide and the HLA molecule in combination. And from here, one of two things will happen. The first outcome is that the T cell receptor does not bind to the HLA molecule. And these are the type of T cells that your body wants. The other outcome is that the T cell receptor strongly binds to the self-peptide that is loaded into the HLA molecule. And this is described as having a high affinity interaction, like a magnet that binds tightly to something that's magnetized. So if a T cell receptor has a high affinity for a self-peptide, that T cell will be deleted in the thymus. And that's because if such a T cell leaves the thymus, it could react with self-tissue and cause autoimmunity. So to summarize, a T cell receptor will physically interact with an enormous number of different self-peptides that are loaded onto HLA. Tight binding to any one of those peptides will result in the death of that T cell. And just as there are many peptide HLA combinations, there are also many unique combinations of T cell receptors. And this part is key. The T cells in the thymus rearrange specific T cell receptor segments to make unique combinations of T cell receptors. Think of the different colored T cell receptor segments as Lego pieces. These Legos are rearranged to make receptors with distinct biochemical properties. I mentioned earlier that T cells in the thymus rearrange specific T cell receptor segments to make unique combinations of T cell receptors. However, this rearrangement does not occur at the level of the receptor. In other words, it's not like there's an enzyme that cuts out a section of the receptor and replaces it with a different section. Instead, this rearrangement occurs at the DNA level, which can be thought of as the instruction manual for the Lego-like T cell receptor. So for example, one specific variety of a T cell receptor, or one family of T cell receptor, has between 197 to 213 different gene segments that can be rearranged to make a unique T cell receptor. And some will bind self-peptides and be deleted, and others won't, which will allow them to exit the thymus and patrol your body for foreign invaders. For example, let's assume that you become infected with SARS-CoV-2. Specific white blood cells, called antigen-presenting cells, shown in blue, will wrap their membrane around the virus, trapping it inside the cell. Here, the vesicle will fuse with the lysosome, which is like a garbage disposal, chopping up the virus and its proteins into peptide fragments. This creates viral peptides that will be loaded onto HLA and presented to T cells. Next, patrolling T cells will use their T cell receptor to fill the peptide antigen that is being presented by this antigen-presenting cell. The T cell receptor will have either no affinity, in this case nothing will happen, or it will have high affinity to the viral peptide loaded onto HLA. If there is high affinity, a signal will be delivered to the T cell that will cause it to what is called clonally expand. So different versions or clones of that T cell will be made. So you can think of this as identical twins of this particular T cell. Not only will identical copies of that T cell be made, but also, more importantly, identical copies of that specific T cell receptor. And now those T cell clones will be able to seek out and destroy cells that are infected with SARS-CoV-2. You might be wondering if T cells that do bind self-peptides can escape the thymus to cause autoimmunity. And the short answer is yes, autoreactive T cells from inside the thymus can escape the thymus. However, there are mechanisms, such as in the lymph node, that can identify these autoreactive T cells and kill them before they can cause autoimmunity. And so the body's ability to prevent immune responses to self-tissue is defined as immune tolerance. So in other words, the immune system is tolerating the presence of self-antigens in your body. Now, immune tolerance in the thymus, which I described earlier, is called central tolerance, whereas immune tolerance outside of the thymus is called peripheral tolerance. The development of autoimmunity is typically associated with a loss of peripheral tolerance due to genetic predispositions, stress, chronic inflammation, or infections, for example. But when you think of autoimmunity, you're likely thinking of autoantibodies that can cause damage by binding to self-targets. And so autoantibodies also play a very important role in a number of different autoimmune conditions. So let's begin by learning about the cells that make antibodies, which are the B cells. And these white blood cells develop in the bone marrow, which is where they derive their name. Similar to T cells, B cells also have a specialized receptor called the B cell receptor, which is an antibody anchored to the surface of the cell. Like a TCR, the B cell receptor also swaps gene segments to make unique combinations that are capable of recognizing foreign targets. However, unlike in the thymus, there's no specialized cells that present self-proteins from every cell type in the body. Instead, the B cell receptor collides with different self-antigens or self-targets inside of the bone marrow microenvironment. And if there is any high affinity interaction, with any of these self-antigens or proteins, the B cell will either be deleted or its B cell receptor variable regions will be rearranged in hopes that a new rearrangement does not interact with that particular self-antigen. This is called central tolerance for B cells. And there are other peripheral tolerance mechanisms that occur outside of the bone marrow, such as anergy, which is a term that's used to describe when a B cell becomes paralyzed and can no longer sense and respond to antigens and make antibodies. Or the B cells can be what's called clonally deleted, or regulatory T cells can help B cells make the appropriate antibodies against foreign targets and not self targets. However, these different peripheral tolerance mechanisms for B cells is beyond the scope of this video. But if you want to learn more about this, leave a comment below and let us know. All right, back to B cells. Now, once a B cell receptor engages a foreign antigen and it receives a proper signal from a specific T cell called a helper T cell, it will also undergo clonal expansion, making more copies of itself and also its unique B cell receptor. This will also trigger isotype switching, where the type of antibody switches from a membrane-bound antibody to a soluble antibody that is secreted from the B cell, where it can circulate through the body and find its foreign targets. So let's tie everything together. The antigens that you're exposed to throughout your life will shape the diversity of your B and T cell receptors in your body. So let's say, for example, that you're infected with a parasite. You will have a subset of B and T cells that will recognize not just a specific protein that is expressed by that parasite, but a very specific region within a single protein. And obviously, parasites don't just have one protein, they have many thousands of different proteins, all of which are potential targets for different T cell and B cell receptors. And so, thanks to clonal expansion, you'll get a population of B and T cells that will expand in number, that will be dedicated to protecting you from that specific parasite infection. Not only in that specific moment in time, but also if you encounter that infection in the future, because some of the T cells and B cells will develop into memory cells that will persist throughout your life. And let's say, for example, you unfortunately develop autoimmune responses that are significant enough to cause disease. So in the case of NMDA or autoimmune encephalitis, you will have a B cell receptor that will target the NMDA receptor that is found in the brain. Or let's say there's a child with pandas. There will be a subset of B cell receptors that can bind the dopamine receptors 1 and 2. Or in type 1 diabetes, you could get a B cell receptor that binds insulin. Or in lupus, you could have B cell receptors that can specifically recognize nuclear proteins such as histones. And so you could see how there could be many different hundreds of thousands of unique B cell and T cell receptor genes in the blood that all have different variable regions. So hopefully now you can appreciate why sequencing the DNA alone of the B cell and T cell receptor genes cannot be used to diagnose an autoimmune disease within a patient without the help of artificial intelligence. Researchers from Stanford University recruited patients with either lupus, type 1 diabetes, had severe form of COVID, were HIV positive, had recently received a flu vaccine, or were healthy controls. Next, they collected their blood and sequenced specific variable regions from their T cell receptor and B cell receptor genes. This consisted of a total of 542 participants with known disease states. They fed this data to an AI machine learning framework so it could learn which patterns of T cell and B cell receptors were associated with specific disease states compared to controls. And after testing this model, they found that comparing 3 variable regions from B cell receptor genes resulted in a best 96% sensitivity and specificity. And then when they compared only the T cell receptor genes, they achieved a best 95% sensitivity and specificity, However, when they combined both the B cell and the T cell receptor gene sequencing into the three different models here, they found that it performed the best with a 99% sensitivity to predicting accurately which disease state that unknown individual had. I want to stress that you won't see this technology in the US or in other countries until more robust studies are performed across a wider range of different groups of individuals. And this will likely involve a clinical trial followed by regulatory approval by the FDA and other regulatory institutions around the world. So this won't come to a clinic near you overnight. And the application of machine learning for immunological diagnosis for other autoimmune conditions and infection associated chronic conditions would also take a long time due to difficulties in connecting geographically dispersed patients with these researchers at Stanford University. In a related note, the Unhide Solve Together Unified Platform is a digital health platform developed in collaboration with patients, experts, researchers, and clinicians. It brings together data from symptom tracking, wearable devices, health history, and validated survey instruments to support patients and help researchers achieve their goals. Now, if you found this video informative, please give it a like, share, and leave a comment below. And make sure to turn on the notification bell for future videos. Bye for now.